Let's start at the beginning with the prokaryotes and early Earth. In this lecture, be able to define gram-positive, gram-negative, peptidoglycan, capsule, fimbriae, flagellum, acceptation, nucleoid, plasmid, anaerobic, and aerobic. Be able to describe the conditions of the early Earth and explain how life may have arisen from non-life. Compare prokaryotes to eukaryotes. That's going to be a bigger goal of this course. Why did prokaryotes come first? And define prokaryote, being able to give examples. The early Earth, well, it was hot, there was no oxygen, and it was not suitable for life as we know it today. But the conditions may have been perfect for life to have arisen. And let's look at how that may have happened. Well, what do we know? The conditions of early Earth were as such. There were very high temperatures. The environment may have been reducing or neutral in regards to the atmosphere, which means there was not oxygen present in its current form. That's right, you couldn't breathe at all. You require an oxidating environment, but the atmosphere of early Earth would have been reducing or neutral. There was widespread volcanism because the Earth was still very young and it was still cooling. The crust would have still been very warm. You probably would have been cooked alive. And there was a lot of lightning. As you may have seen around volcanoes, those discharges of gases do give rise to instabilities in temperature and make a lot of lightning. The fun thing is, these are the conditions where amino acids can form from non-biological precursors, and nucleic acids and lipids may also have formed. Several experiments have been done in this. We can see actually a 1953 experiment we're going to look into a little closer showed that certain number of amino acids could have formed. A higher number was achieved in 2008 when instead of using a reducing atmosphere, they used a neutral atmosphere. What did they do in 1953 then? Well, this is Stanley Miller's experiment. What he did is he used electrodes to simulate lightning. He used boiling water to simulate the heat of the early Earth. And he used reducing gases such as CH4 and NH3, that's ammonia and methane. He used a reflux apparatus, which you'll get to know in organic chemistry, with cold water condensing droplets down and saw if, after a while, there could be gases forming into amino acids. He did show that organic compounds can form from inorganic precursors given the conditions of early Earth. Further research showed that you can take these organic compounds and make amino acids and nucleic acids, even forming polymers. So what use is that? Well, these polymers form into precursor molecules that look like the proteins and the RNA that we have in our bodies. Lipids can form into vesicles, which are similar to our cells in the way that they have a lipid bilayer and they can bud off new vesicles. The formation of polymers is possible when you are putting them on clay, which may have been present at the time. So it acts almost like a catalyst to make polymers from monomers. And some of these polymers are capable of self-replication. When you have something that's capable of self-replication, and you have a vesicle in which it can survive and self-replicate, you create conditions in the vesicle that are different from conditions outside the vesicle and will be more conducive to the self-replication of these molecules. So this is what's needed for life. What would happen if we had free amino acids just formed from nothing now? Well, simple. They would get eaten. They would get devoured by any bacteria. And of course, this is an oxidative environment that we live in right now. So this is not going to be something that happens in the modern day. But before life existed, you can have these precursor molecules formed. We look at this something called the RNA world hypothesis, with RNA forming from inorganic sources and being able to self-replicate via ribozymes, which are enzymes made of RNA. RNA can also catalyze protein synthesis. We see separately the membrane formation, such as those little vesicles, and that can change the internal chemistry and allow new functionality, where step 5, you get RNA coding for both DNA and protein. DNA becomes a template for more RNA. Proteins catalyze cellular activities, such as making more precursors, and, well, that's essentially life. So this is the RNA world hypothesis about how life came from non-life. It came through RNA. And we still see that bacteria use RNA, have DNA, and have proteins. At some point, something had DNA, and it worked a lot better, so all current life is evolved from DNA. What about that early life? Well, that early life with those DNA-bearing prokaryotes formed bacterial mats, and these bacterial mats would grow on top of one another to make something called stromatolites. So the bacterial mats would reproduce and just form more bacteria, and you can see sediment accumulating. Over time, they evolved different methods of utilizing the environment to their advantage, including using sunlight to make energy. And this was the rise of cyanobacteria. We can see that the stromatolites are quite early in the fossil record, about 3.5 billion years ago. We see non-photosynthetic bacteria coming around shortly after, and then we have a debatable form of cyanobacteria starting, and we definitely know they were present around 1.5 billion years ago. Cyanobacteria used sunlight, but they gave off a product called oxygen, and this oxygen was not previously free in the environment. So what does the oxygen do? Well, it reacts with iron. And you get these banded iron formations where you have oxygen mixing in with the iron in the oceans and precipitating out as essentially rust. You can see this in the fossil record of these different layers of rust. And this rust shows that there was an increasing amount of oxygen in the environment reacting with the iron and precipitating it out. Eventually, there was not enough iron in the oceans to take up all the oxygen. The oxygen accumulated in the environment and turned the reducing or neutral atmosphere into an oxidative environment, which killed almost everything on Earth. Yep, oxygen was toxic, but oxygen allowed new types of metabolism to arise, and we can be thankful for that today. So let's look at these prokaryotes. These are the simplest organisms, and the prokaryotes we see today are far, far more complicated than the prokaryotes that would have evolved first. Prokaryotes are structurally simpler than eukaryotes. The prokaryotes do not have a nucleus. The eukaryotes have a nucleus and membrane-bound organelles. Instead of a nucleus, prokaryotes have a circular chromosome. More on that later. And prokaryotes, as I mentioned before, have no membrane-bound organelles. They just have a simple metabolism, and they can grow quite rapidly. So let's look at some things we can tell these prokaryotes apart with. The first off is the peptidoglycan layer. If you have the peptidoglycan layer on the outside, you're going to be a gram-positive bacteria because a certain stain of that peptidoglycan layer is going to show up on the gram-positive bacteria. Gram-negative bacteria have an additional outer membrane that prevents the stain from binding to the peptidoglycan and thus makes them gram-negative. They don't show up because you end up with this stain not staining them. Capsules and fimbriae are things on a bacteria that can attach the bacteria to the substrate. So you can see a bacteria here attached to a tonsil cell by its capsule. It's a sticky layer of polysaccharides and protein, and that prevents the bacteria from just being washed away by the saliva. Fimbriae are hair-like appendages that help the bacteria attach to the substrate, and this allows it to stay in the same location and, of course, reproduce there. If it's a good location, it can reproduce and form a colony. The flagellum is a complex prokaryotic motor, and there's a lot more to cover on this. Your book does a very good job, and I would highly recommend reading it, because the flagellum was formed from pre-existing bacterial cell components, and when they are put together in the right way, this flagellum can allow the bacteria, instead of being stuck to a substrate, to move freely through the water. And that's going to allow it to find new resources. So this was a great adaptation. We call this actually an exaptation because there were structures that were already present in the bacteria that were used for one function, and as they were used for a new function, they become an exaptation. It's an adaptation of things that were already present. We also see specialized membranes in bacteria where we have these respiratory membranes help these organisms utilize oxygen as a final electron acceptor. That's right, these respiratory membranes are essentially similar to the mitochondrion. So these aerobic prokaryotes are basically free-living mitochondria and are able to use things in the environment to make a lot of energy. We also see these thylakoid-like membranes in photosynthetic prokaryotes. That's right, they're basically free-living chloroplasts. We'll get to more on that in another lecture. It's no internal membranes though, just a lot of folding of the external membrane. The genetic material of prokaryotes is held in a nucleoid. This is the region of cytoplasm where the chromosome is actually found, and that single chromosome is actually pretty large, just tightly wound up. The plasmid is an independently replicating DNA molecule that can actually be transmitted horizontally. We'll get more on that in another lecture. We have different forms of metabolism that the prokaryotes have lived in. Basically, if something can be done by a living organism, it is done by a prokaryote. They've had a lot of time to evolve in almost all available niches. Anaerobic bacteria live away from oxygen. These are the ones that evolved first because in the beginning there was no free oxygen. Aerobic bacteria live in oxygen-rich environments like most conditions today. There are also nitrogen-fixing bacteria that can convert N2, just your atmospheric nitrogen gas, which is 70% of our environment, into NH3 or ammonia, which can later be put into amino acids and other nitrogen-binding molecules. So here is a good guide for all the different types of nutrition that bacteria use. You'll want to know these terms because we're going to cover bacterial niches next.